type 1 as the primary disease. So type 1 diabetes is important to study because of the numbers of people that are concerned. We know that the incidence of type 1 diabetes is rising with time, and not only because the registries are better, I would say, and more important, for example, in the southern regions in Africa or Asia. But this is a global rise in type 1 and type 2 diabetes. And for this particular disease, there is no cure. The only possible cure is to replace the deficient beta cells with the stem cell therapy, but the cost is quite prohibitive. Here, I mentioned $500,000 per patient. Actually, we are more above that number now. Type 1 diabetes is a very specific and particular disease, and it's actually a cluster of very different diseases. We always hear about autoimmunity, but this is wrong. This is much more complicated than that. What we know is that there is a decrease in the content of the cells that secrete insulin in the pancreas, which we are called the beta cells. So there are phases of decrease of the beta cells to a certain point where the body is not able to control glucose anymore. So then there is a rise in blood glucose. There is treatment with insulin. So at that time, the patient is symptomatic, loses weight, and have to go to the toilet very frequently. And then when we provide external insulin, exogenous insulin, then there is a decrease in blood glucose levels and decrease in the toxicity of this glucose. And then the remaining beta cells start to work again, and there is a relapse in insulin secretion. And so there is a calming down of diabetes at that point, and that is called the partial remission. So this is something that occurs in 65% of the patients. So this is a story of the beta cells that we know a lot about. But in the pancreas, there are several islets, so it's the little brains inside the pancreas that are scattered in the pancreas. And it's really brains because it's connected to the neural system, and the different cells are intermingled and connected. So the beta cells are connected with alpha cells. And those two cells secrete different hormones, the first insulin and the second glucagon, and they have opposite actions on glucose. So insulin is decreasing glucose, and glucagon is increasing glucose. And what is the evolution of the alpha cells? Nobody knows so far. So this is something that is very important to study. What is also known is that in type 1 diabetes, there is a problem with glucose, but even with the best glucose control that we could achieve with insulin treatment, there will always be an incompressible level of hypoglycemia. So 100 milligram per deciliter is the mean level of blood glucose that is perfect, but due to the variability, there will always be hypoglycemia. And it comes with an array of symptoms, children that have to stop playing, etc., they cannot study, for example, or sleep, with also an impact in quality of life, and a risk of very severe events, because when blood glucose is going down, there can be alteration in the neurological functioning. So here you can see a particular graph of glucose fluctuation in a patient, which has, you can see the gray zone, which is the target range, the normal target range, let's say. It's a little bit broader in those patients than in a healthy patient, but you can see that this patient is spending much time in normoglycemia, with excursions of glycemia going up and falls of glucose going down, hypoglycemia. On the left panel, you can see the mean all of this, and there is a number above of 5.4%. This is the percentage of the hemoglobin that is glycated, meaning linked to glucose, and we all have glycated hemoglobin because we all have glucose in the blood, and the normal amount is 4 to 6%. So it means that this patient has quite a good glucose control, right? And if we compare with this panel on the right, you can see that the situation is very different. Glucose is going very high and is falling quite rapidly when it falls. So if the blue and red lines on the right panel would be a car and the gray zone a road, you can see that the accident is everywhere. But when you look on the left panel below, you can see that the mean of this patient is 5.9% of glycated hemoglobin. So normally speaking, globally and generally speaking for this patient, diabetes is well controlled. So it means that what we are looking at is hidden in terms of the general evaluation of those patients. The problem here is that when blood glucose is falling quite fast, then there is a risk of severe hypoglycemia with loss of consciousness and patients that cannot just function normally or that go into coma with seizures, etc. So this is the definition of severe hypoglycemia. So with our team and in collaboration with other teams and also collaborators abroad, we are gathering samples of patients with new type 1 diabetes and also long-standing type 1 diabetes to study the response to hypoglycemia in terms of alpha cell function and characterize hypoglycemia. So what we first did is to evaluate hypoglycemia. So what is this? Because we all know that there is the number, hypoglycemia, and then the symptoms. But we know that patients are quite differently sensitive to hypoglycemia. So we wanted to clusterize the patients in terms of their sensitivity to hypoglycemia, and we did that with a questionnaire, and we observed that the ones that are very sensitive to hypoglycemia, that have heavy symptoms with quite normal levels of hypoglycemia, are not the ones that are prone to severe hypoglycemia. So this is a little bit different. And hypoglycemia itself, severe hypoglycemia, it's not very well known. So we know that it's a fall of blood glucose, that there are symptoms, but what occurs specifically during the event is not very well known. So my PhD student, Antoine Arvan, studied if there would be a way to predict severe hypoglycemia, because this is very impactful in a child's life. So in terms of numbers, we know that the risk is 1 event every 3 years for a child, but for a teenager, the risk is 1 event per year. So it's 1 event every year for a teenager. So we have teenagers that have several events of severe lows. So they are found in coma in their bed in the morning. So it's quite important to be able to predict these events. So what Antoine did, he looked at the CGM, so all those evaluation of continuous glucose monitoring, and he looked at the days before. He compared the 5 days before with other periods 1 month before, and he cross-validated with very different periods. And what he observe is that severe low is not an isolated event. If you speak to diabetologists, they will tell you, yes, but there is always an explanation. It's a mistake of insulin administration versus a mistake in glucose intake. So the patient did a mistake. He put himself too much insulin and he forgot to eat. Yet, yes, of course it happens, but this is only a portion of those severe lows. So there are changes in blood glucose control before the event, 5 days before, and even we can say 1 month before. So there is a change in glycemic means, in the coefficient of variation, and also time below range. And intuitively, by looking at the CGMs of the patients, it is a long time ago that we thought there would be patterns that led to severe hypoglycemia. And those patterns can be divided in 4 different ways. So that could be a prolonged hypoglycemia after a hyper. So a patient is very high and then spends much time in hypoglycemia. So he kind of uses all the counter-regulatory hormones. He's avoiding his pancreas, let's say. There could be also cycles of hypoglycemia and hyperglycemia. So hypo, hyper, and then the event. Or that could be a very high hyperglycemia and then a very fast downfall of glycemia and then the event. But that was intuitive. We did not know. So we asked Thibaut and his team at Nelissen to study that with AI because that was the best way. So I cannot disclose all of course, the results because this is still pending. The manuscript is in writing. But we put all the items that we could believe that are interesting in those patients' characteristics, level of glycemia. There are at least 48 metrics of CGM evaluation. And so we studied that with the pre-simple component analysis, and we immediately observed that there were items that were falling outside of the evaluation that could be significant in terms of providing and fueling a model for prediction of severe low. So we came up with a model that is able to predict severe lows with a sensitivity and a specificity of about 80%. So it means that the model is really able to predict and to be able to observe the events 8 times up to 10. And the specificity is also very good in detecting negative events. But of course, the positive value, the predictive positive value is a little bit lower. And so the system will give an alarm sometimes for false positive events. But when we look at the graphs, all those false positive events are characterized by hypoglycemia. So it means that when the system is alarming that there will be a severe low, the patient is en route to severe low. So it means that even though the symptoms are not so frank, there will be something happening for this patient. And so we also looked at the SHAP values, which are very interesting to look at what the algorithm is doing, and is taking errors and also messages, very important, and then goes into the predictions. So that is also very interesting to look at the particular values of the algorithm. So what we want to do now, because we have this tool, is to be able to go back and to look at this comprehensive hypoglycemia evaluation. So we have the patients that are very sensitive to hypoglycemia and the patients that are insensitive to hypoglycemia. There are those that are doing severe hypoglycemia quite frequently and patients that will never experience severe hypoglycemia. So we want to do a composite of all of these and to segregate patients with caricatural phenotypes and evaluate the risk of those patients, both clinically with their phenotypes, but also doing exome sequencing, because we would like to look at variants of genes that are involved in glucose control and response to hypoglycemia. So this is a work that is also being performed with the team of Nelissen, and we have the samples. We are ready to go now. So the modelization of hypoglycemia is a composite of different things. Of course, it's insulin and carbohydrate intake, but it's also the way we are able to sense glucose. So the brain is telling us that there will be hypoglycemia, and we are not equal in our faculty to sense glucose. There is also the counter-regulatory hormone, but also the response to hormones, because they have to link to receptors, activate pathways, and this is obvious that we are not equal in terms of metabolism. So we have to take all these parameters into account. So this is basically the story I wanted to tell you this morning. This is the fruit of the work of persons in the lab, the PhDs, and my collaborators. And of course, I'd like to thank the team of Thibaut et Le Put from Nelissen, Marie Bastin, and Cédric Toussaint for their very important and everyday work with our team. Thank you very much.
Thank you, Philippe, for your presentation. Are there questions in the audience? No? It's fun. I throw the cube, and so you can speak in it. No? No question. Yes?
Got it. Thank you for the very interesting presentation. How do you foresee to bring this to the actual usage of the patients? Do you have any like next steps in mind? I think the model will actually make a huge difference to those lives. What do you have in mind to bring this to practice?
There are several ways to go, of course. The issue, I would say today, but this is something that is about to be resolved, as I heard, is that the CGMs are quite few. So the SIMS systems are different from a company to another. So either we do that with one partner or we generalize. And there are also interfaces. So now the patients collect their CGM data based on applications. So it's either in the source or in the reception. It's also possible to have an implementation in terms of pump therapy, because the more and more patients are using pump therapies instead of doing injections of insulin. So they have a device that provides the insulin. So there are several ways. So yes, that will obviously be part of the work of the next years. Yes.
Another question? I have one. Thank you. You just touched upon the existence of abundant, of possibly abundant data from sensors within the patient, so remote monitoring is conceivable. And of course, there is the questions of interfacing those data, etc. From the regulatory standpoint, is it easy or not, or how do you manage to reuse these data which are from everyday care of patients to feed research projects? Is that easy or not? What are the burdens that you have to face?
Yes, well, I think the first step in terms of implementation will be to cross-validate the algorithm in the daily life. So we have, of course, put a lot of efforts and data in the system, but we will have to cross-validate it in real life. And as we said, it's once the work of evaluating the algorithm has been done, the next step is to discuss with the industry and evaluate how to implement that and collaborate. That is for sure something that needs to be evaluated. It's very, of course, difficult, but can go either way as long as it helps the patients. For me, it's fine, and for the team, it's quite fine too. Thank you.
Okay, thank you, Philippe. Now the second talk of this morning will be related to another topic. So the title is Data Science to Support the Development of Bacteriophage Therapy. You may not all know what is a bacteriophage, but Thomas from my team will explain that to you. So welcome, Thomas.
Thank you for the opportunity to present this work. So today I will present how data science can contribute to the development of bacteriophage therapy. And here is what we will discuss. First, I will reintroduce what are bacteriophages, And what is our objective in this project is really to develop targeted treatment using phages for human, but also for veterinary application. And I will present some contribution of data science in this phage therapy development, where we can see how data science can help to have a better characterization of the phage, but also predict its 3D structure and its activity on the bacteria. And this this work is done in partnership with Vesal Biosciences and also with the contribution of the University of Liège, CRAW, the Walloon region, and BOE. So what are bacteriophages? So bacteriophages or phages are simply viruses of bacteria. And these phages are among the most common and diverse entities on the planet. It's estimated to be about 10 exponent 31 phages on the planet, which is more than the sum of all other organisms on the planet, bacteria included. So it's very, very broad. And today it's studied to be used as an alternative to antibiotics, as many patients face multi-drug resistance against bacteria. And so phages constitute a promising alternative to antibiotics in these cases. So our goal is to have a targeted treatment for these humans that have resistance to this bacteria, and develop first in human a phagogram with an associated software. So the idea is start with a patient that has antibiotic-resistant infection, take a sample of the infection, do a culture with that, and use a 96 well plate containing different types of phage and pipettes on each well, a fraction of the sample. And so we will analyze which phase will be the most active against the bacteria present in the patient by using a luminometer. The luminometer will measure the light that will be produced, and there will be a production of light if there is a lysis of the bacteria by the phage. So ATP will be released in the medium, and there will be a reaction, an enzymatic reaction with the luciferin and luciferase, which will produce light. And so in the end, we can select the phages that are the most active against the bacteria. And so the result will be analyzed by the software, and an algorithm will interpret the results in order to have a treatment with the best phages to be used for this patient. There is also a similar scheme that is being developed for veterinary application, but of course with different regulations than in human. So that's the objective. And to help doing this phage therapy, here I will present 3 examples where data science can bring significant contributions. And the first one is to have a better characterization of the phage. Phage have important proteins that are called RBP, for receptor binding proteins. And these proteins are responsible for the fixation of the phage on the bacteria. And this protein will initiate the infection of the bacteria, and they are very important because it's these proteins that will act as the determinant for the host specificity. So if the phage is active or not for these bacteria. So these are very important proteins to be studied, these RBP. But to be able to study them, you have to know which protein are RBP. And so here we use a methodology that has been developed by Bucharts in 2022. And they use data science, they use a model, which is a hidden Markov models combined with a gradient boosting classifier. I will not go through the details. But the idea is that thanks to this model, and simply by giving as input the amino acid composition of the phage protein, the algorithm will tell which proteins are the receptor binding proteins and with a certain confidence score. And this methodology has been internalized on our server to ensure confidentiality. So that's good. Now you know which protein from the phage genome are RBP, so which are the important proteins that you want to study. So now, next point is to study its 3D structure. It's very important to know its 3D structure and study what are the difference between, for the same proteins, but for different phages. So that afterwards, when you have the 3D structure, you can align the 3D structure of different proteins and see what are the difference within the same species of phages or between different species of phages, and so that you have a better understanding of the phage, which can help you in choosing the best phage for your patient. And here we use AlphaFold. So AlphaFold, it's an AI system developed by Google DeepMind to predict the 3D structure of a protein, given its amino acid sequence. And we internalize AlphaFold on our server at the Analytics, again to ensure confidentiality. And so as an output, you have a PDB file that describes the 3D structure of the protein. And you can now visualize it. Here is an example of a visualization of a protein using, for example, an R package, which is called NGL Viewer. And so you can have an HTML file that you can visualize, make rotation, and see really the 3D structure of the protein. Doing the collaboration with the University of Liège, we were very interested in two specific RBP, which are the short tail fiber and the needle really at the bottom of the long tail fiber, which are very responsible for the attachment of the phage on the bacteria. And so you can see the representation using AlphaFold on the bottom left for the short tail fiber. These two proteins are in fact trimers, and so you can visualize multimers using also AlphaFold. And here you see that the three monomers are really parallel to each other for the short tail fiber, whereas for the needle, they are each monomer is folded to create really this needle and have this receptor at the tip of the protein, and this is the receptor that will fix to the bacteria. But then you have the visualization of your protein. You want to compare the same protein but for different phages to see the difference. So you need to align these 3D structure together. For this we use another tool, which is Molstar. And you end with a file where you see really the alignment of the proteins and what are the differences between the proteins that are responsible for the difference between the phages. And you can measure the difference, so how far are they from each other, structurally speaking. And for this, we use the RMSD score, which is the relative mean square distance, which is the sum of the square distance of each atom with the two structure between being aligned. You can measure really the distance in the 3D space. You sum the square of this distance, you divide by the length of the protein, and you take the mean square. And you have the RMSD, and then you can visualize all these RMSD if you have the same protein but for a lot of different phages, and you can create heat map, that's the figure on the right, and you can see that for some phages this protein is very close from each other. That's the case in blue, and in red they are more different from each other. So that can give you information of which protein is important and which phase you should choose. And the last contribution of data science that I will present today is how we can use data science to predict the activity of the phage on the bacteria. So here we want to predict the lysis activity of the phage on different bacteria. This is traditionally done using EOP for efficiency of plating, which measure the numbers of lysis plaque of the phage on the bacteria layer. And then there is a cutoff which say basically if the phage is active or inactive against this bacteria. But the method is quite long. It lasts 24 hours, and it's difficult to do it many times in parallel. So in Vesal Bioscience, they wanted to replace this technique by another technique using the luminometer that I spoke earlier. So in the luminometer, you will have a 96 well plate where you will put the phage with the bacteria. And if the phage lyse the bacteria, they will have release of ATP, which together with luciferin and luciferase will produce light. And so you can measure the activity of the phage by measuring the light that is produced using this luminometer. And so we want to be able to predict EOP using these value from the luminometer, which has the RLU for relative light unit. And we do that for 2 different phage species, which account for 9 phage together. And we want also to give a certain probability of activity, not only to say if it's active or not, but with a certain level of probability. So to do that, we had the data of the standard method, EOP, and the RLU with the luminometer for 9 different phages. We did some feature engineering. So we noticed that we had to standardize the RLU with a standard, because there are fluctuations of the RLU from experiment to experiment. And so that's why we created this ratio, having the RLU with phage divided by the RLU without phage, so to have a standardization. We inverted this ratio and then calculated the ratio after lysis versus the ratio before lysis, so to have a measure of activity of the phage on the bacteria. And we use a model, which is a generalized linear model with these features. And then we apply a sigmoid on this core going out of this model to give a probability of activity of the phage. We noticed that we could fine-tune the model per phage using an offset, and we validate our results using tenfold cross-validation, and we did a test on a separate dataset. And here are the results on the bottom right for the validation. So on the x-axis, 1 means that the phage is active from the EOP method, -1 it's inactive. And on the y-axis, you have the probability of activity giving our model using RLU. So if you are above than 0.5 probability, you give the prediction that the phage is active. If it's below, you give the prediction that the phage is inactive. And so all the points in green are correctly classified given our methodology. Compared to the EOP, the points in red are wrongly classified. So if you look on the test results on the bottom, you see that we had very good results globally with an accuracy of 91%, so meaning that 91% of the phage were correctly classified. So that was the results with the phage, again, Pseudomonas aeruginosa. We did the same against Staphylococcus aureus for 4 phages, and here the results are a bit lower, but still satisfactory, with an accuracy of 76%. So to conclude, phage could serve as an alternative to antibiotics for patients which have infection that are antibiotic resistant. And here I just show you some examples where data science can contribute in this phage therapy to have a better characterization of the phage with this identification of receptor binding proteins, which has very important proteins to be studied. Also, it's important to study the 3D structure of this protein, visualize them, see what are the differences between the phages, and finally be able to predict the lysis activity of the phage and the bacteria. These are only some examples where data science can contribute. As a perspective, more and more we speak about generative AI. There is a possibility that generative AI also help in this therapy, where we can imagine that in the end it will design the whole phage genome, and so you can synthesize it using cell-free technique. That's a promising possibility. So I will end here. Thank you for your attention. Thank you for the AI Week for the opportunity to present, and thank you to Vesal for the partnership. Thank you.
Thank you, Thomas, for the presentation. Are there questions in the audience? No? Well, maybe I have one. So we saw also in the previous talk and in this one that Medtech innovation requires a lot of different expertise. You have to interface with hardware, develop software, analyze data with more traditional or more recent machine learning approaches. How do you manage with your colleagues to organize, to be able to deal with all this variety of projects and technologies?
It's important to have a good collaboration, I would say. So here we had this collaboration with Vesal Bioscience, who could bring all their scientific expertise on phages. We had also the contribution of Liège University, which has a very deep understanding of the phage and the study on the receptor binding proteins. Here in the analytics, we are here to put everyone together and bring the data science field. But indeed, we need scientific expertise to combine these data science technique with relevant science.
Okay, thank you. Another question from the audience? No? Well, thank you again, Thomas. Now we'll switch mic. We'll get to the third presentation of this morning. And so it's nice because then we have one presentation from the Brussels region, one from the Baldwin region, and then one from Flanders, and so that's good. So Wad, I invite you to get on stage to present your topic. So Wad, as I said earlier, is co-founder of MoveUp. Thank you, Wad.
Thank you. It's a pleasure standing here. So indeed, my name is Wart. But I'll actually bring a bit the story of Philippe. Because I'm an engineer, and I've met Philippe about 10 years ago. Philippe is doing orthopedic surgeries, hip and knee surgeries. And I've met Philippe, he's doing his practice at Marmille-Lares. I've met Philippe during an MBA. And me with an engineering background, not in healthcare, we started to talk. And I was talking about that we were putting with Philips at the time, we were putting TVs in the market, which we could monitor, which if they had a bug, we could upgrade remotely and even prevent some of the bugs which could happen in the field. And I was asking to Dr. Van Overschelde, I asked him like, and how do you do that with patients? And then it was silent. And then I said, wait, we're not monitoring health data yet. Okay, this was 2015. So I fast forward a bit during this presentation. I hope I'll inspire you with some real practical examples of what we're doing and what we started. But before I really go to the AI particular modules which Philippe is now using in his practice, This is kind of what was the moment where I had kind of, okay, we need to do something about this. Here you see a timeline of about 2 to 3 months, a typical surgery or other intervention. So the doctor has a very good knowledge about, of course, the patient when they go into surgery. But then between that surgery, between the discharge to home and the first consultation or even the second consultation, there's a lot of blind spots. I think the first presentation already mentioned, like, okay, there's now applications or wearables which have the possibility to actually cover in those blind spots. But of course, a lot of things can go wrong during those, let's say, really after surgery. You can have high pain levels, your wound can infect, but also maybe your rehabilitation is not going as expected. So we said, you know what, if we could actually improve already the insights, maybe we can also control how the patient is evolving after such an intervention. And this is actually what we started to think about, to brainstorm about, but also in a combination with, let's say, an engineering side as well as a doctor side. We said, okay, let's build this and let's really start to have our first clinical studies, first clinical trials. Those were in 2017 with patients. What you see on the graph are an example of the type of data we're collecting. Really, initially, it was important for us to collect pain levels, but also activity levels. But you'll see we, in the meantime, have evolved to collecting about 400 different data points for a standard care path, which we've developed. Now, one of the main questions we asked ourselves is also, if we bring these type of technologies, how to motivate the patient, because you can, of course, ask them a lot of information, but if they don't see the benefit, then it's going to be hard to keep on having that information. One of the elements was definitely building an application which is low barrier. If you think about hip and knee patients, you can think about elderly patients. Average patient is about 65-year-old. By the way, some numbers in Belgium, there's about 50,000 of these type of surgeries happening on a yearly basis. So that's a lot. It's also an extremely, I would say, successful typical surgery. So patients are pain-free again, they're functional again, they can even go back to sports. For some of them, depends a bit on the profile. But it's important to know that in that sense, if we want to interact with the patient and have those insights, you need particular engagement, like information modules, but also you need to collect data, which are sometimes very subjective, but also we connect with wearable devices, which are objective data. Now, this is from the patient perspective. And if you look how we can also, let's say, this is a bit where the engineering side comes in, how, let's say, we've started to build continuous feedback loops. On the one hand, if the patient is reporting that information, there is some simple data processing going on in the cloud, which is depending on the progress of the patient already giving feedback. For example, if the patient has a specific question about going back to work or starting to drive with a car again, these type of modules can be activated on the spot for that particular patient. But also if they have suddenly high pain levels, which at the moment, for example, 3 to 4 weeks after surgery, this is not normal, well, then there's particular triggers going back to the patient or even to a care team, and this is a second feedback loop which comes into play, to the care team, which on the spot can actually monitor remotely patients on a day-to-day basis. Now you might think, okay, this is going to add a lot of work. Well, on the contrary, because this can be done on a bigger scale, this also allows to only, in that sense, get an escalation of the patients who at that moment need extra attention. And this is where, again, that on the right side of the on the left side, sorry, of the slide, you see the treating physician, the surgeon coming back into play, where, if necessary, of course, a physical consultation can again be planned in. On the bottom, you see a screenshot of how a typical dashboard to follow up the patient is looking. Now, if you are able to do this on a patient-per-patient level, so think about having all that 400 different data points per patient, you can also start analyzing how, more like in a descriptive analytics, I would say, how the different patients are doing. On the left side, you see age distribution. On the right side, you see, let's say, different daily data which is being reported. It can be, again, activity. It can also be symptoms, pain levels. In the middle, you see a patient-reported outcome measure. You see two groups being compared. And this was actually also for us kind of an aha moment, especially also, for example, for Philippe. But also, we're working together, for example, with the societies of Belgium, but also international societies, to start collecting on an aggregated level and to see whether you can see difference between two types of interventions, or maybe because of comorbidities. And the forgotten joint score is one score which tells you, after 6 months after surgery, did you forget in the meantime that you have a new implant? And that's one of, let's say, key patient-reported outcome measure. And here we're comparing a group A versus B, green versus blue. And built in, there's also a statistical analysis already possible. And this is kind of moving, and there's still a lot of hospitals in Belgium, let's say, collecting this type of data in Excels, or even only, or not at all, or even only during studies. Well, now you have a prospective real-world data collecting platform which allows doctors, clinicians, but also researchers to start collecting this data and immediately do further actions or decisions based on that. Back to Philippe. How did he now start combining these data insights in his daily practice? If he gets a patient with a specific, let's say, set of complaints, before he even sees the patient, he asks them to fill out actually a certain assessment, an assessment score. Here you see on the left side an LLM-based interaction, which is asking the patient, I tried it in this case, when, for example, what are the typical symptoms they're experiencing? When it started, whether they're taking any pain medication for that, when, for example, the symptoms are getting worse, when they are relieved, what it is hampering now, whether they already did any other surgery before. And on the right side, you see then the structured, and of course this is not in Fire or any coding yet, but just this is in that sense human readable for the doctor to analyze. On the right side, you see a summary of what happened with that patient. Depending on the results of this structured data output, we've developed the MoveUp index, and this is giving a score between 0 and 100, which is actually allowing the doctor to decide between a conservative treatment and a surgery. And that's really important because this is, on the one hand, combining, let's say, information about historical data, comorbidities, but also particular symptoms and the situation of the patient, together with also X-ray analysis. You see in the middle on the right side, you see also a full X-ray analysis, which does first landmark detection, and then it starts also to detect, for example, the degrees of arthrosis. And what you see on the left side in the score is a green score, which means, in essence, this patient would be a good fit for surgery. Now, there are still, and this is, in that sense, the white box speaking, there are still red flags, so contraindications to operate on this patient. You can read them for yourself there. But there are also green flags. There's elements which are justifying, let's say, to go for surgery. And on the right side bottom, you even see for this patient a kind of how we expect that the milestones, so for example, how satisfied they would be when they would be able to stop pain medication, when they would be able to stop crutch use, or resume back to work. So that information is being able to combine that information at the moment of consultation. Now, where is the data coming from? In the meantime, so 10 years ago, I've met Philippe. 10 years fast forward, there's about 25,000 different patient and the decision makings being logged in the platform, which allows us to bring this clinical decision support tool into the platform. Then the moment of decision is taken, the surgery is being prepared, and then actually application is being used to, the patient application is being used to kind of form a bit of a context around the patient and also prepare that patient for surgery. Then on surgery, this is courtesy of let's say, the typical medical device companies who develop implants and robots during surgery. So this is a screenshot of one of the surgical planning tools, in this case of Microport. So these are tools which Dr. Van Oversjelden is using. But also on the right side, you see one which is imageless. So the left side is the robot. On the right side is an alternative, which is imageless, which is actually using, the doctor is using augmented reality glasses to, at the moment of surgery, define the right angles, the right cuts to actually place the implant during surgery. So those are two examples which are used during surgery. So once the patient has received the surgery, we go to, in that sense, the recovery phase. Patient is discharged from home. And that's, of course, the moment that we are applying, on the one hand, the application, but also we apply video-based analysis of the patient. You see on the right side on the picture a range of motion exercise which the patient needs to do, which is then analyzed and then can remotely follow up the extension and flexion of the patient. And then finally, last but not least, there is also a long-term follow-up possible. So here, if the patient would see back, if the doctor would see back the patient at 2 years, he would see a full report completed. On the left side, you see the patient-reported outcome measures. On the right side, you see the little blue balls which you see on top of the box plots, is actually where this patient is compared to a cohort, compared to other cohort. This can be specifically the doctor's benchmark, but this can also be the full, let's say, worldwide database of these type of surgeries to be able to compare where this patient is. On the bottom right side, you see the whole, let's say, daily data which has been collected on this patient with, on top of it, annotated the different milestones which this patient has been able to achieve. So that kind of closes, let's say, the typical workflow of the doctor. Now, needless, I think it's also important to add that each of the components are connected. And it's only a step by step that we're able to do this connection, which, for example, means that the surgical planning, so how the implant would be placed for a particular patient, that data is now finally being connected also with, for example, the electronic health record, with the MoveUp database, to kind of make sure that each decision which today is being taken in a primary, let's say, use case of the patient can also be reused for future patients, similar type of patients which are being treated, and where, for example, today we make a decision, or doctor makes a decision, we can also measure the context on how the patient was evolving, and we can reapply that on new patients coming in. So this is kind of really important that we see this arrow going back, that we can close the loop in that sense to make sure that also future patients can rely on these type of insights and improvements and predictions. Something has gone wrong here with the green bar in the copying, but that's okay. I think I've talked about providers, the doctors themselves, but also the patients. We also see that in that sense for example, for implant manufacturers, but also for biotech and pharma, these type of real-world data collection can bring huge insights on how the product is used, but also increase the product adherence or their own product adherence in the field. As well as for insurers, if we look to the efficiency gains which can be taken during the decision process by the doctor. For example, I was talking about Philippe already a lot of time, but the product is being used in Belgium, in neighboring countries as well, in the US and Australia. And we see that actually by putting this little effort to start applying the way of working, they also immediately see, in that sense, the terms of benefits in the decision making, but also efficiencies in the outpatient care, so the moment the patient has gone home for example, rehabilitation. Obviously, we are a product on the market, which means that we comply to medical device regulation. Also, if we look to the amount of, let's say, studies, both real-world as well as prospective studies, we've been able to do and publish, those are really important steps to bring these type of, in that sense, innovations to the market. So I would also say, and when I was discussing with Thibaut, he was asking to me like, okay, bring a real case example, which is already being used in practice. I would also say there's a lot of elements still cooking in the kitchen, in possibilities like bringing, for example, digital twin elements, or even a continuous and interactive chatbot of the patient, with the patient to help them to rehabilitate. Those are all things which are cooking in the kitchen, but we cannot just release it as such and test it out, because we're talking about a very regulated market and product here. In practice, we, on the right side, you see some of the regulatory elements we comply to. So there is quality management system, also IT security system, and of course, I've mentioned the medical device regulation. And in the US, we're registered as a software as a medical device with the FDA. And I think it's also important to understand that the principles we've been able to apply in orthopedics, they can also be applied in other domains. And this is what we see more and more. And that's why it's also interesting in that sense about the first speaker, thinking like, okay, how to bring this into practice, because we've been able to multiply this also in other domains like obesity surgery, cardiology, oncology. And we do think that we're still, in that sense, scratching the surface in terms of how this type of insights in primary and secondary use can benefit the patients and the doctors' lives. So thank you very much.
Thank you. I'm going to this side of the room to motivate other people to. Thank you, Joy.
So thank you for the presentation. I have a question. You spoke about the integration of the data with the electronic health record. So what is your experience with this integration with the hospital and maybe also with the GP?
So it's, thank you for the question. So it's not easy. Let's start with that. I think one of the elements which made a change for us, let's say if I talk about the Belgian landscape, is that in 2020, our application for orthopedics got the reimbursement, one of the first reimbursed applications in Belgium, which meant for us that actually we had suddenly kind of the support of RISIF INAMI, and also a lot of hospital directors understood like, hey, this is an application which is not only validated, but also reimbursed. And that opened for us the door to discuss, for example, with a Nexus Health or with an Epic or with different electronic health records in the hospital to start connecting. So that's one element. So we are connected, and we also see that this is a must at one point. I don't think that all solutions out there from day one can have that connection. But there is a very nice initiative which I'm supporting together with, it's also one of the data capabilities, FOD Santé initiatives, which is the telemonitoring prescription initiative, which is putting a digital hub or a data layer between the electronic health records and actually digital health solutions like ours, which is really important to kind of make sure that not each of the initiatives like us needs to go individually to all different hospitals, but actually can make sure that there is one layer in between to connect to. This is the hospital connectivity. You also asked about the GPs. The intention is in a phase 2 to make sure that there is also connectivity to the GP systems. I would say not as interactive yet as with the hospitals, unfortunately, but at least, for example, through COSO, that at least GPs can have the report which I showed to the patient, that they can have access to those type of reports and know also which type of care pathways or care plans are active for the patients which they have in front of them. So it's still step by step, and I hope also with the European health data space that this type of interoperability, well, I don't hope it will for sure come there, that with the EUHDS, will even be accelerated for the future.
Thank you. Another question? Yeah, I'm not a very good thrower, so.
Thank you. Thank you. It's a related question. So let's suppose that the patient is going to another place and doesn't want to work with you anymore. Will it be possible for him to have the data, or how is it working?
So the typical, let's say, if we apply the GDPR principles which we've worked out for this type of collaborations, we're typically a processor on behalf of the hospital, so of the doctor, to process the data. But of course, there is the moment that the patient is being, proposed to work, for example, with the application or the digital data interface. There's in that sense a consent, an informed consent, which links, in that sense, the processing of that particular patient. Also, on the one hand, make sure that everything is being well understood and being agreed for the processing for different purposes. And those can depend and are depending on the use case an opt-in or opt-out. So there's a primary use of the data, which is really for that particular preparation for surgery and post-surgery treatment. And then there is currently it's an opt-in for secondary use of the data or for scientific purposes. Does it answer your question?
Well, my question was very simple. Can the patient have the data or not?
Yes. Well, it's kind of, we also comply to the GDPR rules in that sense. At any moment, the patient can request to retrieve all the data. Or even ask, in that sense, the removal of the data, or we provide that data in a, in a transferable way if the patient wants to use that data in another system. We are, and I think that's maybe looking a bit to the future, but we are part of a consortium, an EMEC ICON consortium, together with the EMEC and ID Lab, to actually convert some of those data storage into patient-centered health data vaults, solid-based. So that, in that sense, your question is more, I would say, answered in a different way. That in that sense, the patient is the owner of the data by default through their pods. If you know the technology, feel free to come to me if you want to know more. And they're actually sharing their data with different healthcare professionals or industry, more, prospective rather than now is the other way around.
Yes, as you probably well know, in the academic setting, one of the biggest issue we have with patient data is the management of confidentiality, informed consent, and GDPR. It's very difficult even for data that we already have in medical records. For example, every time we want to evaluate something, and even prospectively, of course, it's becoming even more complex. We have to anticipate everything that will be performed with the data, not even evaluation, but also sharing, etc. So can you comment a little bit on how this is managed with the platform?
I think the part of the answer is maybe also in which I gave to the sir in the back, which is we have the different consenting upfront already with different purposes. The moment actually the patient is being onboarded, initially, of course, it's for primary use. The patient goes to a particular treatment or a diagnosis. So that's a very logical moment to say, okay, I consent on using my data for these purposes. And then we have the flexibility at that moment to already ask, like, okay, can your data be used for scientific purposes or for research? The interesting thing is also the moment that you have that, let's say, digital link with the patient, there's also possibility to later on in an, I would say, quite low threshold way to go back or to recontact with that purpose in mind, the patient or the citizen, maybe they're not a patient anymore, but a citizen, for, let's say, a study which you maybe didn't foresee 5 years ago, but to go back to the patient and, let's say, re-ask consent or to put it as part of an ethics committee, for example, to get that approval or get that reapproval for that purpose. So I would say it lowers the barriers. It's not yet the best thing to do. I think if we talk about patient health-centered type of data pods, there I believe it's more logical to, it more directly goes to the patient as their own owner of the data. But of course, that's also not something which happens in let's say overnight. So it's going to be a hybrid system for a long time. But at least we see that it helps because we're collecting that data, can be used for real-world data and real-world evidence purposes from the beginning. But happy to talk still further about some of those, how that things works in practice.
Thank you once again. So now we've seen a set of examples in medical technologies where prevention, detection is really important. Now we will go to the second part of this session, which will focus more on biotech and in particular in the field of manufacturing. Once we discover, invent new treatments, we have to produce them. And as I highlighted in the introduction, this is also a place where digital innovation can play a role. And so for this part, we will have two presentations with two different speakers. First, we will have Pascal Yerkens, Principal Scientist, Drug Substance Innovation at GSK here in Wavre. Presenting an actual case of application of modeling to a specific step, but a crucial one in the manufacturing of vaccines. And then we'll have a second talk by Jean-Louis Jouve from Teraxel, who made the trip from France. Thank you, Jean-Louis. To explain, to report a work that has been made to see how as an industry, we can integrate this kind of innovation into environments that are subject to good manufacturing practices. But so let's start with Pascal. Pascal, welcome.
Hi everybody, thank you for this invitation. Yes, I'm working in the development of new process vaccines. Just two words about the vaccines. The vaccines, as you know, it's a product that helps you to protect you against a pathogen. And for that, the goal is to inject in your body. For the old technology we inject inactivate virus or a piece of bacteria, but in new technology we just use one protein from the pathogen. And we have a new innovation in our field. It's called the mRNA. We just inject the code that produce the protein of interest. But for all the vaccines, the base is always to use cells. And for that, we use different kinds of cells. We can use mammalian cells, insect cells, yeast, or bacteria. We first start the production by increasing the number of cells. And when we reach this number of cells, higher number of cells, we can start the production of the antigen. And during the development, we have different constraints, but the first one, of course, is to increase the productivity of the protein of interest. But as the product will be injected into the people, we have also a lot of concern about the quality. And in quality, we have different topics. One of them is the purity, the final purity of the product. And for that, after this different step, we add different steps that we remove contaminant coming from the cell culture. But we have also we need also to better characterize the protein of interest. For example, we need to determine the composition of this protein, the folding, the shape of this protein, and so on. But the problem when you are working with the cells is the complexity of this organism. For example, you have an example here on the picture. This is a simplified view of metabolism, and in order to well understand what's happened during the production of the protein, you need to follow the evolution of this metabolism. The traditional approach that we use up to now is during the production of the protein to take samples at different time points and to make a lot of offline measurements. It works quite well, but the problem is you have a fragmented view of the process. And also, as you need to take samples, you also take the risk to, for example, contaminate your product. Since a few years now, we have access to process analytical technology. This technology allows us to have a view in real time of the evolution of this metabolism. For that, we put into our bioreactor a probe that will take spectral measurement. And thanks to different machine learning algorithm, we can convert this spectral measurement into value. And value that we can have in real time. I will illustrate this concept by just one example. I asked my colleague in production to take sample for me, and they accept to take one sample every 4 hours. But as we are in the GMP facility, they only have the possibility to just follow the evolution of the cell density, the biomass. For the same group, we create different chemometric models by introducing a PET approach into this facility, and now they have access to much more information than in the past. If you see the blue dots correspond to prediction made by the PET model, and in red this is the sample that I took manually, and in order to compare the prediction to the real world. With the PET approach, we can have access to different parts of the metabolism and also to see the evolution of all these components in the real time during our production. What can we do with that? But we will use this new approach to take this decision. The principle is the following: we have the bioreactor in which we make the cell culture. In this bioreactor, we have a probe that are directly in contact with the product, with the cells. The probe will take spectral measurement in continuous mode. Then every time they finish to take a spectral measurement, they restart a new spectral measurement, and so on until the end of the cell culture. And thanks to a machine learning algorithm, we can convert this spectral measurement into value. And you see different kind of value. Then we have different kind of machine learning algorithm. Based on this prediction, we can do 2 things. One is the monitoring, and the second one is the feedback control loop. To illustrate the monitoring, I put this example. Just for the moment, look at the graph on the left. You see blue dot. Blue dot is coming from the prediction. We take spectral measurement and convert into the value. And red dot, this is the sample that we take manually to make offline measurements. And you see the point that I already mentioned, that we have a maximum in the blue dot that are completely missed by the red dot because we are not there when the peaks happen. If we this kind of process should be repeated during the time, and if we accumulate enough data, we can define an average comportment for this kind of specific metabolic. You see the green line, and we can, this is average, and we can define upper and lower limits that allow us to see when something is fail. You see on the left graph that at a certain time point, the blue dot go outside the red limits. At this time, we can launch an alarm, for example, to say to the people, process people, oh, be careful, something happened, do something. Okay. But here we still need to have the intervention of human people. The evolution for us is to go to the digital twin. The digital twin is, we use a model that will mimic the normal comportment of our process. And the goal is to use the PET to check in real time the comportment of the culture to the model. If the two values are the same, we do nothing. If there is a difference, we can launch different action that will give back the process into the normal condition and control the productivity. In conclusion, the introduction of this technology in our process, but first we can improve our knowledge about the process. We can improve our way of working, but also we can improve the final quality of our products. We also can facilitate the decision during a process, and we can do that in real time. And we minimize the risk because we don't need to take a sample anymore. And also by using a digital twin, for example, we hope that we will decrease the number of fail and increase the number of batch that are going until the end of the production. The challenge for us, as mentioned, we will collect a lot of data. That means we need to have a system that manages this huge quantity of data. We have also concern about the calibration and validation of the machine learning models. We need to prove to the authority that our model will make the good prediction, and also we need to think about the life cycle of the model. In 5 years, in 10 years, we need to prove that the model is still working and give the good prediction. We have also the concern about the integration of this kind of technology in our facility, because we need specific IT architecture in order to collect the spectral measurement, make the conversion, and send the information to another system. And of course, we need specific profile for that to operate and maintain the systems. That's all for me, and thank you for this invitation. If you have questions.
Question from the audience? No? Yes.
Thank you. Thank you for the talk. I'm interested in the PAT. It's kind of innovative technology, right? Innovative? No, my question was I would like to ask. Well, I'm not really from this field, but I feel that the AI technology in general are taking a lot of rules more than just this, right? Is it true?
information, and then when we have the information, we can take the decision, you see. But for the moment, the machine learning is just used to convert the spectral measurement into the value, and after that we will use probably other kind of model to make the decision automatically.
I have another question. Where do you think what's your perspective for this AI technology in drug development? Let's see.
Yes, for us, the goal is to better control our process, of course, and to have a fine view of the evolution of the process. Our target is to improve the quality of the final product, of course. And yes, we would like to automatize more and more the process in order to have a better product at the end.
Everywhere in this life cycle. Okay, thank you.
I just have one that will make the link with the next presentation. So without entering into confidential details, so you mentioned that, of course, these innovations have to meet the expectations of regulatory agencies. What kind of aspects are there really attentive to in the integration of that kind of technology? Can you share a bit your experience?
Yes, according to the first discussion that we have with the authorities, the main challenge is to prove the performance of the model and to show that this performance is constant along batch after batch during the time, and to propose a strategy to check this performance and how we can prove that the value, the prediction that are made by the model is the good one, and that's why we need to have a mix between offline measurement and inline measurement from time to time.
Thank you. Thank you again, Pascal. So in direct connection with this talk, a presentation by Jean-Louis Jouve from Teraxel on how to integrate data-driven innovation in this kind of GMP environment. Thank you, Jean-Louis.
Thank you. Thank you, and first of all, I would like to say how pleased I am to be here today in Brussels. And we have several topics to mention today. The first, of course, is to present who I am, because I saw in my slide presentation function. So my function is I'm an IT compliance expert in Teraxel. But I'm not here for Teraxel, but for A3P. A3P is an association in France, also in Belgium, in Switzerland, and in many countries. A3P is an association that focuses on aseptic processing for pharma, biotech, and others, and medical device as well. And we have a group called e-compliance. We call group GEEK in French. We are working on the regulations evolution. We are working also on the evolution of technologies, and we're trying to match these two aspects, of course. Our topics is really related to GMP, so I'm not going to talk about clinical trials or laboratory experiments or even pharmacovigilance aspects. As you can see, we have a group of people that are also experts in different companies. We have a mixed culture in A3P, which is mixing industry companies and also suppliers of these industries. So Thibault is a proud member of this group, and I appreciate his presence, very useful. We will talk a bit about what we mean by regulations, related to AI. I will not go into very much details about that because it's a bit labor intensive and it will take a lot of time, but I will try to summarize the working of the groups related to mainly 3 documents that we have been using. The first one is published by the LEM. LEM is the union in France on the pharmaceutical industry. They have published a document last year and very useful for inputs because it has been also reviewed by the regulatory agency in France called ANSM. And second document is FDA. We are really keen on checking what's going on the other side of the Atlantic, especially in this area. And we have some interesting documents submitted for review and for comments we are working on now. And this document is really useful because it brings some interesting insights for the application of AI in the regulated domains. And the next one is a document we are going to publish really soon now about the same topics about the LEM, but giving more practical details on how to implement AI in a regulated environment, regulated GMP environment. Okay, first talk is about computerized system because we are discussing about systems. We are talking about regulations, and we have some documents already existing on the regulations. The main one being the Annex 11 for Europe, and this is describing how we can implement a computerized system in a regulated environment. So we have this introduction that tells you, well, we have a computerized system. What is a computerized system? For the regulations, these 2 things. First of all, the process, which is controlled by the system, and the system itself, the computer system. So altogether, it forms what we call the computerized systems. So when the regulations look at the system, they don't look only at the system itself, the hardware and the software. They look in more global way as integrated with the process. So there's something missing in this nice drawing. It's called data. Data are absent from this nice drawing. In the regulations, we don't. In this specific regulation, we don't focus really on data. We have another chapter 4 documentation, which is related to data. And these 2 documents, chapter 4 and Annex 11, are currently being reviewed. And we expect some changes in these documents, specifically based on new technologies. Now, for the AI system, there's the big difference because everything starts with data. So we have a first process, which is a learning process, building the model based on existing data, learning inputs and outputs, and so giving some representation of reality. So we learn from data. We have new techniques, machine learning, deep learning, and we can build a model which will represent reality with some probability, of course. It's not 100% probability. So we have this specific process that we need to tackle in order to implement systems in GMPs. Again, when we speak about implementation in the regulations, we have some specific steps that are already described in the regulations. We call it system lifecycle, and here we have this representation of the lifecycle based from the GAM5. GAM5 is also a big document not a regulated document, but an industry standard that describes the implementation of systems. So we have several steps, very traditional for computerized systems. We call it concept phase, we have a project phase, operation, and so on. We also have consideration for the retirement of systems, because when we take out the systems, we need to keep track of all the records from the system. So when we move from a traditional system to an AI-based system, we have a different life cycle. And we have in the A3P group, we have worked on a specific life cycle, which I will now describe a bit. The concept phase is very important because we need to define what we want to do with the system, specifically what we call the concept of use, use case basically. We need to, of course, have all the data collections. Do we have the data available for this implementation? Once we have defined all these items, we can move to the project phase, which involves data preparations, with all the different steps I will discuss later on. Important steps with the data is to split the data into groups and data sets, what we call the training set and the test set, and we will later on implement these different data sets during the model optimization, model fine-tuning, and also the testing of the model. Then we can move the model in production. Then we will have some performance monitoring. This is something we have been discussed before. The system will be checking actual data, and we can optimize the model with this new data, this new actual information, and we can loop back into the process, into the project phase, to perform the optimization of the model. So this model, in fact, starts with a business case, as I mentioned. This is an extract from the FDA document I mentioned before. Very important is to define the question of interest and the context of use of the AI implementation. We have discussed a bit some different approach. One approach is to have a fully automated system without any human interaction. Of course, this process is very important and critical because there's no other way to make sure the system works if you have no human to supervise the process. For example, we have in GMPs, we have examples like visual inspections. You know, we produce vials, for example, and we have to check if there's no particles inside, and so on. So this could be automated. So there's cameras making images of the vials in different angles, and we provide some AI analysis of the images, and we can give the answer: is the vial without particles or not? So that's a very integrated process in the production of drug products. In other cases, we have no automation. The AI system is just providing information that a human will use or not, depending on circumstances. So this is a very different topics in the implementation of the model. And that leads to what we call the risk assessment of criticality assessment of the system. Again, from the FDA, they differentiate what they call model influence, which is the contribution of the AI system to the decision or to the production of information from the process. Because you can have AI system plus manual systems, for example, that we still provide similar information. We have also the consequence of the decision. The impact in GMPs is monitored on several aspects. The main aspects is patient safety, which is linked to product quality and also to data integrity. So we have these three items to consider when we implement such a solution. I will now try to focus on different aspects, critical aspects of the implementation. The first one is qualified personnel. When we implement a system in development, or also when it's in production during the run phase, we need to have qualified people around the system to make sure we have the right expertise, and it's critical also for AI system because data scientists, I think we have this discussion before, data scientists working with business people, process owners, all together in order to make sure the system works correctly and we have the right answer on the questions we ask for to the system. This is also described in what we call the URS, User Requirement Specification, which is describing in more details what we expect from the system. We need to make sure we have the right, all the items covered. On the right side of the, on the slide, you have the more details that will be also described in the documents I mentioned before. But typically we have to make sure we have all the data, the type of data we are processing are accurately defined. We have considerations also on the infrastructure. For example, we have in the previous presentation, we have this, the importance of the feedback of the process has to be very quick, and it's the same case for visual inspection. So we need to have very specific hardware specifications and so on. So we have a first step, which is very important. We focused on the data as a starting point. In the groupment A3P, we discussed about a concept or acronym called VERIDIC. VERIDIC because we need to have enough data to make sure this project is working. We need to have accurate data, exact, which will represent the things we want to measure. Representative also, integrity is also very important in pharmaceutical industry. We have documentation on the data to make sure the objects are we understand the object that we are manipulating. We can industrialize this data. Sorry. We have a comprehensive understanding of this data as well, and also we have to be ethical. Well, not so much in GMPs because we're not talking much about personal data, but we have to consider, in some cases, of course, GDPR and the AI Act, which is applying. More information about the data. In fact, we need to make this process transparent and auditable. To summarize, we need to make sure all the data we are collecting and processing, we have a full audit trail of the data changes, the cleaning process, things like that. All the processing we do on this data, we need to have a full control in the development process. Also, this is an interactive modeling process. We understand that it's very hard to find the right model the first time. So there are some interactions, and we know that also from a regulatory point of view, if we have this kind of process, we need to build some transparency again in the process. So everything that's manipulating data, all the decisions that are made on the models, the fine-tuning, optimization, and so on, has to be transparent and has to be documented. Qualification activities, well, it all depends on the criticality of the process of the system we are going to implement. It's driven by the risk assessment, but the typical items are, of course, installation qualification, operational qualifications, and performance qualifications. That will refer to the different aspects. Installation refers to hardware, OQ refers to the functional performance of the systems, and the actual performance monitoring relates to the live system. Performance monitoring is when the system is in production. We have the system working. It's operating on a daily basis, and we need to monitor the system to make sure it's not derivating. There's no bias involved in the system. There's no drift in the model. So we have to make what we call periodical review of the system, and to make sure that it's still complying and the model is still valid for the process we are monitoring. So as a summary, the important thing are the identification of the use case, because that will trigger the whole assessment process and the whole qualification process. We understand also that we have a holistic process in the implementation of these systems. So we have a way. We need to have a way to have a full documented approach in this validation process. Documentation, of course, I mentioned that, and we have something we brought up during these discussions in the JIC meetings: attention to operators and people using the systems. For example, we had one use case which was implemented in Sanofi. It was automation of PQR, product quality review, which is also an annual review. And we had interesting information about the AI providing some conclusions, let's say, on the different aspects of the product quality review. And we have this information about, is the information changed by the expert or not? And we understood that at the first implementation, we have 80% of the comments coming from the output from the AI, which was changed or modified by the expert. And we understand also that in the coming years of implementation, they are really focusing on what has been changed and why it has been changed, because people feel that AI implementation, operators or users may tend to rely too much on AI conclusions and not or apply their critical thinking to the conclusions. So this is something that's interesting to discuss maybe in the future. As a conclusion again, Annex 11 and Chapter 4 will be revised in the coming month. We expect some changes on the new technologies, especially AI. So we are waiting for that and waiting for comments on that as well. Thank you very much for your attention.
Thank you, Jean-Louis. Are there questions? Yes?
Thank you. if I read the conclusion, but also your explanation, it kind of reminds me a bit of the medical device regulation. Even did you look into links? It talks about intended use. It talks about risk-based assessment. It talks about documentation, auditability. It talks about continuous, let's say, re-evaluation of what you're doing and how it's being used and whether there's any issues. Can you comment on that?
Well, you need to know first that the regulation on computerized systems has started from the medical device. The first issues were related to X-ray administration systems that were deficient, controlled by computer, and the impact on the regulation was started from medical device and shifted to the pharma industry. So, of course, in the pharma or biotech, we are focusing on drug substances or drug products, not directly with the patient most of the time. But yes, we are working on similar concepts. So the regulation, especially in the FDA, has moved from the medical device group to the pharma group with similar concepts and similar processes. Yes, absolutely. And it's still the same bit in France, in Europe as well. Yeah.
Thank you. Another question? Jouve, maybe can you comment a bit on what kind of persons should be involved in that kind of validation project? Is it something that is confined to the quality assurance department, or how do you see it?
Well, it takes some expertise in several aspects. It involves some technical knowledge, of course, but also some, business and also quality assurance principles. So it's really a group of people working together. Unfortunately, validation has just been associated too much with documentation, which is true, in fact, but we are working also in the GEEK A3P to make the change on this a bit. But yeah, it involves lots of working together, understanding the system, and making the right documentation, basically, the right records.
Thank you. And a last question? No. If not, as everyone was very kind in respecting the times allocated to each presentation, we have the time to make a 5 to 10 minutes break and then get back for the last part of this session. Thank you. welcome back. I hope you have had interesting discussions during the break. There will be opportunity to discuss more just after this last part of this session, which is yet on another topic in healthcare, which is more the management, structuration, and use and reuse of medical practice data. And for this, as announced, we will have two talks. A first one by Dr. Thierry Klein from Solstice, who will provide a concrete example of the use and how to make data interoperable across multiple hospitals. And then a second part from Andries Nelissen from FPS Health here in Belgium to discuss how, among other topics, how the EU health data space will be integrated in Belgium. So please, Thierry, you can join me on stage, and the floor is yours.
Thank you, Thibault. So I'm working as consultant in Solstice, but I'm also working in a hospital and a group of hospitals. And so my topic today is to show an example of transformation of data from text to structured data by using artificial intelligence. And so I want to thank also the different partners. You see the logos of the partners. And first of all, the name of the project is DPI for INA. I will explain rapidly what it means, because DPI, of course, is Dossier Patient Informatisé in French, and INA is Institute of Analytics for Health. It's a big name, but it's a local initiative in Wallonia to put some database, home database in hospitals to reuse the real-world data. And so we choose to add a layer to this first project, and the layer is to use the text in the electronic patient record. About 80% of the information within an electronic health record is text. So you can imagine it that this source of data cannot be forgotten. And we choose 3 use cases, mainly the two first use cases, allergy and vaccination. I will explain later on why. And also a little part of oncology is just the diagnosis in oncology within the text, just to see and to recognize the diagnosis in the text. Of course, the workflow is quite typical of such a project. So the first step is to extract the data from the electronic patient record. It's mainly medical reports, such as discharge letters, consultation reports, and so on. And then it's not written here, but the topic of the day is innovation versus regulation. And regulation is quite important because we have to cope with pseudonymization of data. For instance, the typical number of patients, what we call NIS number in Belgium, you cannot put that in the database. You have to generate a pseudo key. It's okay. It seems simple to do, but it's not, because you need a third party with a service to exchange, and an API to call this part, and so on. So that's the first part, but it's not the only thing to do. The second challenge is to say, okay, but there are some personalized data into the text. So you have to also clean the text, and we are using, for instance, an open source software developed by APHP in France, and we use this program to identify and to hide some personalized data into the text. So I have no time to explain all those details, but it's quite important to emphasize that we took more than 6 months to do that, and it was not foreseen. So we took the time to do that properly before analyzing the data. And then the second phase is natural language processing tool. I will explain it later on. It's an LLM, a large language model that we used. And we are going forward with the SNOMED-EYE. So SNOMED-CT is the interoperable classification. And more than classification, it's quite a language, coded language for medical information. And then we put the file care set. What's that? It's just for the first use of data. We had to exchange medical information between the practitioners. So it's the first primary use of medical data, and the secondary use of medical data, it's OMOP. OMOP, it's a standard of database to research, and that's the two direction of our project. So that's all the image of the project. So you can recognize we start from medical data, and one important thing in the project that we want to compare the data from the hospitals and the data from the first line general practitioners, and it's quite difficult to do that, but it's quite important. And we took the three UCS, allergy, vaccination, and oncological diagnosis. And as you see in the schema, we process the data, and we put the data into OMOP database, but also into a file repository in order to exchange the data with other practitioners in Belgium. There are, I would say, the two main goals: primary use of data and secondary use of data. But the hospitals, it's a network of hospitals, MOVE, is CHC Montlégia in Liège, and also Saint-Vith and Eupen. And they decide and they ask to us, yes, but that's fine to collect the data in a relational database, but we want to use it internally to manage the hospitals and to have some request on the data. And so this is the way to put a clinical data warehouse. And there is the third workflow is an internal hospital use of the data. Just as an example, if you see in a record, a medical report, okay, the patient is allergic and he has rhinitis due to pollen. Okay, that's quite obvious in the text, but it's information with a lot of attributes. And so we decided together to have a common logical model of data. And it's not only, okay, one information in text and one code. Certainly not. You understand in the phrase, you know that you have an allergy and you have a manifestation. There are at least two codes, but it's not enough, because is that now, is that in the past, is that severe, is not severe, is that, I would say, who says the allergy? So it's important to say, okay, I am allergic to penicillin. Who said that? Is that my grandmother says that in an emergency room? It's not the same if a specialist in hospitals has proven that this allergy. And so, as you see on the left slide, we put a common data model. I have written here, I would say in SNOMED CT, but you can read it easily, I think. The first is, okay, allergy to what substance? Okay, is there manifestation or not? Is that a real allergy or an intolerance? Is that severe or not? And then you have a lot of context data, and we want to code all those data too, because we have to cope with, for instance, there is no allergy to penicillin, it's proven. So the negation is classical in NLP, we have to cope with that. And also if we say, okay, but the mother of the patient has an allergy with penicillin, it's also you have to code that. And also, I would say the specialty and the temporal context is that in the past, is that now. And the last one, it was added by an exchange with a specialist in the hospital, and he say, okay, one really important thing in allergy is to say, who says that? Who says that? And so we add and we code the person or the qualification of the persons who signs the allergy, because it's not just that allergy to penicillin, it doesn't mean anything. And so on the right side, you see, for instance, how we can code the information, and to the left, we see entities, relationships, classification from NLP. And then we translate that in SNOMED CT, in a template in SNOMED CT. Template is the logical model with all the attributes. And at that point, we had a problem. We faced a problem because we have some medical knowledge, and so we wanted to interpret the data to say, okay, but it's probably not correct, the data, I will correct the data. No, we stop and say, no, we don't have to interpret the data. Garbage in, garbage out, okay, maybe a little bit garbage, but the specialist has said, one information for me is to compare, for instance, the prevalence of allergy in penicillin in my hospital versus in literature. And that's the important output is, okay, the information is not correctly mentioned in the electronic patient record, but it's important to reflect exactly what it was said and not to interpret the information. This is the first reason. And the second reason is it's not possible to interpret if you automatize the workflow. And we want to semi-automatic code of all the information, because there's a huge mess of information of report, and so we cannot put a terminologist on the screen for each expressions. So, in a nutshell, what we have done today, we have extracted medical reports from 3 hospitals with different medical software. We de-identified the data, and not only the pseudonymization and cleaning the reports, the content of the records. We have an experience of rapidly increasing of performance in LLM. And so at the beginning of the project, I worked to validate some results of the NLP LLM. And they say, okay, yes, that's an allergy. No, that's not correct. That's a correct manifestation or not. And then they say, our colleagues say, no, stop it. No, we have tried Llama 3.1, and then the Llama 3.3, and the performance and the quality of the result of the NLP was better and better. And so today, we are using Llama 3.3, and that's quite good. So it's like a revolution for us in the project. And then we are, I would say, We are in the middle, and we want to automatize the process in order to be able to code a lot of data, and not manually, but by computer and software. And one of the challenge, which is not yet okay, it's the coding, the automatic coding. But through the LLM, it's coming, and we hope that we can say, okay, this is NLP, we have information automatically coded in SNOMED CT, and then it's quite automatic. It's always semi-automatic because you have to validate the data at one point. And so, once again, it's obvious that the quality of the data is quite important, because if you transform the data, you will introduce a bias, and you will reduce the granularity of the data from the doctor. And so the clinician has to validate at each step of transforming data. One important decision in our project is to say, okay, we have to push the data to exchange, for instance, allergic data for the patient between hospitals and general practitioners. That's one goal. So we can take the data and push the data into FHIR standard. It's exchange standard in healthcare. But we decide not to do this way, because if you push the data directly into this format, you lose information, you lose granularity. And so we decide to have an intermediate XML file which keep all the details we were able to code. And this XML neutral format, as the name neutral, it means that we can push and we can transform this XML file into different directions. FHIR is for the primary use of data, but also OMOP to open the way to clinical research. And the other decision, quite important, is, and it's not yet finished, it's to compare the data from the hospitals and the general practitioner, because it's quite interesting to compare the two for allergy, vaccination, and oncological diagnosis. Thank you. On the road to new adventure, I hope that Nick Rodwell, the responsible of Tintin trademark, will not pursue it, but as a result of the project right now, we want to go further, because it's a first step. We can do that, but we have to more and more develop some real research cases, the use cases, and also to real exchange the data between the hospitals. We have proven that it's possible. We have some deliverables that can be used with other hospitals in Belgium, thanks the FOT, the SPF program data capabilities. Andries Nelissen will speak a little bit more afterwards. And so I want to thank also the Ministry of Health permitting us to do that project. Thank you very much.
Thank you, Thierry. Has anyone a question for Thierry here? Yes?
Thank you for the presentation. My question is, why do you already de-identify in step 2? Because I assume the whole processing is done locally at premise, or is that not correct?
That's correct. In fact, inside the network of hospitals, because there are 3 hospitals and different use case research, they decide to anonymize, no, not to anonymize, they identify all the data from the beginning. Just extract the data and directly all the data were pseudonymized. And so they can go back if they need all the information about the patient, or if they need the information to, I would say, to put that in a real clinical database. They can do that because they have the mapping key between the pseudo key and the real key of the patient. But at the beginning, they wanted to protect the data, even if the database is in the environment of the hospitals.
And then maybe a follow-up question. Does that also mean that if, for example, if I would be a specialist, for example, oncologist, would I already see the result of your structuring in the electronic health record, or is that only, let's say, in that separate database which has been structured and in that sense processed?
A quite good question. In fact, they are moving from one system to another for the electronic record, and the goal is to have a neutral, independent OMOP database, but with possible, with, I would say, SQL possible from anyway, software, and the future electronic health record. It's open, and DNA Analytics has developed a special software for the clinicians to see the data.
Yeah, as there are 3 hospitals, and you said that you are using an LLM, NLP and LLM, and so it needs large computer capacity, I suppose. And so do you build that in the 3 different hospitals? And 3 big machines in the 3 hospitals?
Thank you also for the question. No, because they wanted to gather all the information in one network hospital server. And that's not so easy because, as you know, the network of hospital is quite 3 separate hospitals. But they decided to put all together the data, and that's the reason why they decided to pseudonymize all the data. And so they just need one infrastructure with GPU or CPU software, hardware installed. And now it's possible to get some infrastructure within the hospital, not a little one. So Saint-Vit and Eupen is not possible. They don't have the capacity on the staff to manage that. But CHC Montlégier, it's possible. And so they decided to put the infrastructure and the system only in Montlégier, but in the frame of the network of hospital, MOVE network.
Thank you. We'll close this now to go to the last presentation. Thank you again, Thierry. Thank you. Now we welcome Andries Nelissen from FPS Health to discuss the EU health data space in Belgium. Thank you.
Thank you. I will discuss the European health data space, but maybe not directly in Belgium, but the big picture of the European health data space. So it was started, the first presenting the project in 2022. And in the beginning, it seemed like very impossible to reach an agreement between the member states. But finally, today, beginning of March, the EUHDS was published. So it is really like a major milestone for all the European member states to have this European legal framework in place to enable seamless health data flow for the benefit of us all. So let's go to the most important aims of this regulation. So it wants to empower individuals. They want to foster a single market and enable trustworthy data use. So empowering individuals is enhanced digital access for patients, for citizens, but also for healthcare providers. Foster a single market They want to have a unified market for EHR systems and AI and promoting interoperability. So we are defining the implementing acts so that we have some standards for the EHR vendors. And of course, trustworthy data use to have a reliable framework for using the health data for research, innovation, but also for policy making. The EUHDS is based on two key infrastructures, My Health at EU and Health Data at EU. So My Health at EU is already a voluntary system where some member states have a national contact point, and they exchange already some patient summaries of e-prescription. And health data at the EU, it has to be built, and they are defining it for the moment to make sure that we have a seamless health data exchange between the different member states, so that we can also use this data across the different borders of the European member states. So this regulation really focuses on the safe and secure exchange of health data while still holding up the EU data protection standards, GDPR. So it is really based on the GDPR. It will not replace the GDPR. It's also linked with some other legislations, the Data Governance Act, the AI Act, etc. A lot of European regulations go together with the EUHDS. And of course, citizens will always have instant access to their own digital health data, and health professionals will access medical records for treatment across the borders. So each member state will have to use the same format, and it has to make it available for other professionals in other member states. And so we have a common ground for assessing this data, so that we can also, and also we have introduced some innovative ideas like digital testing environments to test the EHR vendors, and also trusted data holders to try to simplify the process of getting a permit to use the data, etc. And also we have defined some governance rules for the health data, but still we have a balance with the competencies of each member state, so they also have some significant control how data will be processed within a member state. But still, there is a harmonized framework across all these member states. And therefore, because we have this harmonized framework, the EUHDS can unlock research potential with anonymized, pseudonymized data based on health records, but also clinical trials, even genetic data, and also other data categories that are defined in the European Health Data Space Regulation. And so that will support innovation for cancer research or treatments or rare diseases, etc. So we really believe that will be a very big game changer for all innovation in Europe. But there were some challenges, and the EUHDS hopes to overcome these challenges. And I've summed up 5 important ones. And you have, of course, first of all, data availability and access, but also data quality and standardization. I will go into deeper detail later. You have regulatory and legal barriers, transparency and trust, of course, and then the end, but not least, of course, governance. So data availability and access. So there is a big challenge to make this data a high quality health data available, and also to have this in the same standard, and also to oblige the data holder to make the data available. First of all, you need to have the high quality data, but then make it available so that you can do some research with it, or population health management for governance. And therefore, the EUHDS had a solution that we have My Health at EU, have health data at EU, the infrastructure to make this sharing of data secure and trustworthy, and also enhance the pool of data available. So therefore, data holders will be obliged to disclose this electronic health data, also for secondary use. This for primary use, there will be obligation, but also for secondary use. And also the data disclosure will be through a defined, harmonized structure. It can be direct or via a health data access body who will deliver a data permit to the data user who asks for the data to the data holder. So there are also some safeguarding measures in place to protect intellectual property and trade secrets. Also some articles in the regulations defined to also give these guarantees to make also innovation sustainable. Then data quality and standardizations. So to improve data quality and interoperability, the EUHDS mandates some common standards and certification for HR systems, but also for secondary use, there will be data quality labels that will be defined so that you can have an idea what data sets are, what kind of quality standards, etc. Then thirdly, on a regulatory basis and barrier, so now you have each member state who has its own competency for healthcare in each member state, but still we try to create a harmonized legal framework so that this health data sharing for secondary use can be possible, so that we can overcome the differences between member states, and so that we can ensure for the patient that the data will be exchanged trustworthy, secure, and also very efficient, so that the data you reuse for research and policy making can have a larger pool of data, while still guarantee the security of the data. And the EUHDS regulation was published beginning of March, 5th of March, but it will enter into force into force on the 26th of March in 2029. And then 2 years later, we will have to exchange the first priority categories for primary use. It will be patient summary, e-prescription, and e-administration. Then again, 2 years later, it will be other priority categories. And also at the same time, we also have the data available for secondary use. And so there's a time frame between 4 and 10 years. 10 years, it means that other parties can be involved outside of the European Union. Transparency and trust, of course, very important. We have to give some trust to the patients, to the citizens. And so we have, with the European Health Data Space, a framework that ensures this trust by giving some definitions and regulations for the data access. And also we will align with the GDPR and other regulations. There is the right to opt out for secondary use for these health data. Member states can also go further. They can provide additional safeguards and even stricter measures for some data categories. For example, human genetic data. There are some stipulations that member states can really restrict this, and that can even go further than only opt out. And also for primary use, member states can also decide to provide an opt-out mechanism or not for the patients. Of course, governance, very important. Now we have a system where some data, some patient summaries or e-prescriptions are exchanged over the borders within the European Union, but with this regulation, it will be obliged. So it is no longer a voluntary system, it will be the obligation, and therefore you need a strong governance. So that we have some boards for primary and secondary use, but we have a unified governance for the European Health Data Space regulation. And of course, the success of the implementation will rely on the cooperation between the different member states to address the challenges mentioned, but also to have this consistent regulations and interpretations across the borders of the European member states. So we should not minimize the importance of governance in this matter. And also because member states still have a significant control, they can add some additional safeguards for HR vendors, for certain data categories, for certain implementation where there will be some minimal possibility to have adapted to their own situation in each member state. Then I would like to make the link with the AI Act. So I think there's also the same principles that you can find back. You want to ensure the safe and trustworthy development and deployment of AI technologies. So for instance, in healthcare, it's very important. You have this classification of AI applications in function of their risk levels. And also we want to promote innovation with the EUHDS, but also with the AI Act, and also addressing certain ethical concerns. So I see some synergies between the EUHDS and the AI Act. you have this regulatory synergy that both framework focus on trustworthy, secure digital ecosystem. The EUHDS protects sensitive health data, and the AI Act ensures that the AI systems using this data meet also some safety, transparency, and ethical standards to make the chain of security complete. Of course, context matters. When you're developing AI system, you have to take into account the population where you are developing your system, and so you have to use the data from this specific group to have accurate and safe performance of your AI algorithms. Quality is key. High-risk AI systems require high-quality, error-free, and representative data for training, validation, and testing. So EUHDS is also working on that to make the data available, making a larger pool of data available, making the data more quality, following more quality standards, etc. I already spoke about about governance for the EUHDS, but also you have data governance. I think we should use the EUHDS as a lever to augment the data governance capacity within our ecosystem, especially for healthcare. It's a very big challenge for them. And so we can avoid that the biases and gaps are represented in the data sets. So fighting bias is very important. And also we want to advance healthcare innovation. So the EUHDS-driven access to high quality data will empower the AI Act to act full and to have some more groundbreaking solution, improving the outcomes for patients and also improving medical research so we can fight maybe rare diseases or cancer, etc., other things that are still complicated to solve. Let's go to some concrete things that we are doing in Belgium. Thierry already talked about that we had this data capabilities program. We have also the innovation program. And so we want to continue on that. So we want to promote intra-hospital data innovations to improve this data exchange between healthcare providers. For innovation, we had 3 teams defined. We had, of course, AI, this is the third one, AI development, but we also had integrated care and also the task distribution and skill utilization in healthcare to optimize that and with the use of data. So these projects for the innovation, they have a time scheme of 1 year. We invested this last year 20 million. They have a time frame until the end of this year. And we selected 26 projects. And we have 86 hospitals participating in all these projects in the different regions of Belgium, and more than 40 partners that will be working together with these hospitals. So we really have a large scale and a lot of partners involved. So our idea is, okay, we want to have some implementations in the hospitals. Then at the end of the year, we will push for scale. Already now we will push for scalability to have a significant impact. And then in the end, we want to scale this up to all the hospitals in Belgium, even the smaller ones, so that they can profit from the innovation done by the larger ones. I will not have, my time is limited, so I will not go into the detail of some projects I selected about a specific use case on AI, but I want to talk about some potential risks for the hospital sector. Of course, human resources is very challenging, and it's very difficult to keep talent within a hospital. It's also difficult to work together with these external service providers, with procurement, also legislation. IT, there are some delays in the development. As also, you have also strict regulations, anonymization, pseudonymization, respectation of the GDPR, etc. Of course, government needs to deliver on time. It's not always that easy if you are working with innovation. Expertise is required. It's very difficult to keep them, and also requires additional resources. And then in the end, how will we really make it sustainable, and how will we upscale it, and will we have enough financial means to make it happen? These are a lot of potential risks, but still, I think we will learn a lot from these projects. This is my last slide. So we have some good practice already in place. We have some more collaboration between hospitals. Very important that they are really working together. They have to work together and to connect, to share these resources and the expertise. So this capacity and knowledge building is already happening. They will also have better project governance, have some better skills, know how to manage so quite difficult, important projects. We are already doing some documentation of the lessons learned, so that we also can, of course, share it with the others, so they make it implementation even easier. And then these IT documentation that has to be more open source, so that it can, some services can be reusable for the hospitals. Bigger hospitals, Jerry already told about it, you have a network, there's a big hospital, they can invest in a large computer system or investment, small and less, so maybe they can reuse the same building blocks that are made by these larger hospitals. Thank you very much. There's a lot of things to do still, but thank you for listening, and hear you soon.
Thank you, Andries. Maybe we have time for one question. Yes?
Yes, thank you. Very interesting. My question is about the EUHDS. So it will be officially, I don't know how to say in English, but en vigueur from 25 of March, so in a few days. But I was
29. It's March 2000. Sorry.
Yeah. So that was my
29.
so that was my question. No, okay. Legally complicated, but exactly, precisely, there is, as far as I know, nothing before March 29, and this is totally crazy. So, for example, in my opinion, for example, the right for citizens to have access to their own data only in 29. So can you confirm that there is absolutely nothing before 29? And is Belgium going to, let's say, follow rules who are there already before or not? Thank you.
So I think it's a big process that has to be done. So we only have 2 years to be ready for the EUHDS. So it will demand a lot of effort for a lot of parties, for the government, for also the EHR vendors, and also the data holders. So you still have the general data protection regulation that's in place, and if to have access to your data, you can use the legal articles in the GDPR to ask your data portability, etc. So it's not only the EUHDS that's regulating this issue. But what we want is that if you are exchanging a patient summary, that the patient can have access to this patient summary for the secondary use, that there's a opt-out mechanism possible, so the patient can decide if you want to make this data available for secondary use or not.
We'll have to discuss this afterward if you have more questions. So, yeah.
Thank you.
Once again. So I will be very brief in concluding this session. So once again, thank you all for being present here or online. There are apparently even more people online than here. Thank you for the tech team, BOSA and AI for Belgium, for setting up this event. In summary, I think that we have seen that there is a high motivation and a high potential in the healthcare sector to use digital technology, data science, and AI, and that the field is quite already used to integrate new regulations for many years now. So we see that the AI Act, the EUHDS have been now agreed on. Now it's the concrete implementation of those measures that will impact the field. And so probably an attention point for authorities is to look at the details of how this will be implemented, not to put too heavy burden at every level of the field, on academics, small company, large companies. And my last message is to thank once again the speakers of this morning. I think it was very varied and interesting. So thank you all.